Okay, it's my pleasure to welcome everyone to the fourth webinar, the APS COVID webinar, today given by Professor Arup Chakraborty, and then moderated by Professor Mehran Kardar. I would like to give a couple of updates, just so you know. Many of you, I'm sure, have been on the previous 3 webinars. I apologize for the last minute cancellation that we had 2 weeks ago, obviously affected by the elections in the US. As you know, the series is organized by the CRRG group. So if you have any questions, send an email. We always like to hear from you. Today, I also like to share a few updates for those who are regular to our webpage. You might have noticed that it has changed a little bit. I would like to also kind of like focus your attention on 3 specific links that you have. The first one is here, where it's a link to receive information and alerts for upcoming COVID webinars. The second one is the webpage about the COVID Research and Resources Group. And finally is the link to join the CRRG. The reason why the first link is important, because we're transitioning to a different way of announcing webinars. Traditionally, we were using multiple channels, but we tried to make it simplifying. So please go to this link. This is merged with other APS webinars, many focusing on careers. That's why a little bit strange choice. But if you go to the bottom of the list of different webinars series you can join, there is the COVID Research and Resources. Just click there. And that means that you will be on the email distribution list about future webinar updates. The second link is the COVID Research and Resources Group. So this is a web page where we try to post some of the most important announcements that are happening. And then finally, if you join the Engage, which is open to everyone, there is instructions how to join. Then you also get access to other announcements, as well as then discussion groups that we have. For example, from the last webinar, we posted all the Q&A with questions and answers, etc. So I would also like to say that we do have the APS YouTube video channel that's specific to COVID. And that's where you can see all the previous lectures that all the previous speakers graciously allowed us to share with broader audience. We have 2 more webinars planned for this year, both in December, so in 2 weeks and then in 4 weeks. And then we'll kind of like have a break for winter and then resume probably in late January with the same way as this time. We'll send another request for feedback after next webinar. We would really like to hear from you, especially as we're planning the future webinars, the topics you would like to hear. Obviously, there's a lot of exciting happenings that are happening now with some of the vaccines getting available. So everybody is very excited about that. With this, I'd like to stop sharing my slides, and I would like to invite Dr. Mehran Kardar to introduce our speaker of today.
Thank you. I'm delighted to introduce my friend and collaborator, Arup Chakraborty. Some 20 years ago, Arup recognized a promising moment in time with the field of immunology that called for overarching conceptual frameworks. He concluded that the vast body of empirical observations, advances in basic immunology, and the ability to collect large amounts of new data can be combined with appropriate tools from physics and other physical sciences, engineering, to address important critical health problems. By developing models based on statistical physics and studied by state-of-the-art computational means, Arup embarked on a quest to provide testable and quantifiable insights into this multifaceted system. Computational immunology is now a growing field that spans several disciplines. Arup was a pioneer and is now a leader of this new trust that combines methods rooted in physical sciences and engineering with experimental and clinical immunology. Through his insights and the many collaborations he has formed, Arup has provided the conceptual frameworks for understanding the fundamental mechanisms that underlie how the immune system functions and how harnessing that knowledge can address medical needs. His work has not only led to insights and advances in basic immunology and infectious disease, but has also inspired new directions of research across basic sciences, engineering, and medicine. I'm looking forward to hearing Arup's presentation.
Hopefully you can see my slides. Can you?
Yes.
We can.
Thank you very much, Robert, for the invitation. And thank you, Mehran, for the overly generous introduction. So the human story is inextricably linked with infectious disease-causing pathogens. Our immune system combats these infectious disease pathogens upon natural infection. Vaccines have enabled us to win many battles with infectious diseases. Vaccines modulate our immune response in ways that confer protection from infection. So let me begin by saying that we have an adaptive immune system, which is remarkable in that it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes, including those which may not have evolved when we were born, such as the SARS-CoV-2 virus, which causes the COVID-19 disease. To roughly see how this works, consider infection with a virus. When a virus infects a cell, it hijacks the transcriptional machinery of the cell to enable synthesis of new viral proteins and the assembly of many new virus particles that can go on to infect other cells. B cells are important players in adaptive immunity. We have about 10 to the 11 B cells, and most B cells have a receptor on their surface that is distinct from the receptor on the surface of another B cell. If the receptor on this particular B cell can bind sufficiently strongly to the spike proteins of this particular virus, biochemical reactions cause it to get activated, it begins to proliferate, and then a Darwinian evolutionary process occurs, which I shall describe a bit later, which ultimately leads to secretion of soluble and even higher affinity forms of this receptor, and that's what we call antibodies. These antibodies can then bind specifically and strongly to the spikes of this particular virus and then dispose of it in a number of different ways. But antibodies principally target free virus particles in blood or extracellular spaces. Now, some of these proteins of the virus are chopped up into short peptides, about 10 amino acids long, and displayed on the surface of infected cells in complex with human proteins called MHC proteins. We each have 6 to 12 types of these MHC proteins, but there are thousands of variants in the human population, and different MHCs tend to bind somewhat different peptides. T cells are also important players in adaptive immunity. Again, most T cells have a distinct receptor on their surface, and if the receptor on this particular T cell can bind sufficiently strongly to this particular viral peptide, it gets activated, starts to multiply, and then can coordinate an immune response in a number of different ways. For example, one kind of T cell called CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it can secrete products that will kill this infected cell and the virus particles that they harbor. Now, after you've cleared an infection, most of the B cells and T cells that proliferated in response to this particular infection die. But a few remain as so-called memory cells that can mount robust and rapid responses upon reinfection with the same pathogen. This pathogen-specific immunological memory is the basis for vaccination. With a vaccine, you try to induce memory B cells, T cells, and antibodies that are specific for the bug against which you wish to protect the host. Now, the SARS-CoV-2 virus, like other viruses, infects human cells by using its spike protein. In particular, its spike protein binds to a receptor on human cells called ACE2, which plays a role in regulating blood pressure, and is extensively displayed on cells in the lung, intestine, heart, kidney, etc. And that is why this virus can cause quite a bit of damage. Now, after the spike binds to the ACE2 receptor, another protein on our cell surfaces called protease, depicted here as scissors, cuts the spike protein into two, which acts like releasing a spring, which then forces fusion of the membrane of the virus in our host cell and entry of the RNA genome, thus causing infection. All current efforts on developing a vaccine against SARS-CoV-2 are focused on eliciting antibodies that can bind to the spike protein of this particular virus, thereby preventing it from binding to ACE2 and causing infection. The Moderna and Pfizer vaccines are based on delivering the RNA corresponding to this particular spike protein into our cells, which then makes the protein, and then antibodies develop against it. The Johnson & Johnson and AstraZeneca vaccines are based on using an adenovirus to deliver the spike protein's DNA. And the adenovirus normally causes mild disease, but in this case, it is engineered to be non-replicating, and its genome has an insert, which is the spike protein of the DNA. So again, this virus infects our human cells. The spike protein then is made by our cells, and antibodies develop against it. As evidenced by recent results, we anticipate that these vaccines will be highly efficacious. And a part of that reason is that the spike protein, or its entire genome, of SARS-CoV-2 is not mutating very much. But imagine in the future, if another virus, which causes a respiratory disease, evolves, whose proteins are highly mutable. Now, vaccination has saved more lives than any other medical procedure, but we have not been very effective in designing vaccines against highly mutable pathogens using the current paradigms. HIV is a prominent example. We don't even have a universal vaccine against influenza. What is required in order to eliminate scourges like this from the planet is rational design of vaccines based on a mechanistic understanding of the pertinent immunology and virology. The challenge of high mutability is illustrated here for HIV. In these phylogenetic trees, the number of N's corresponds to the diversity of circulating strains. And you can see from this diagram that the diversity of flu strains in the entire world in a particular year is comparable to the diversity of HIV strains in a single infected individual, and completely dwarfed by the diversity of strains in just one region in Africa. This high mutability presents a major challenge for vaccination because HIV can mutate to evade vaccine-induced antibodies and T cells that lie ready and waiting for certain strains. So today I'm going to briefly describe some of our work concerning both the antibody and T cell arms of this challenge, beginning with our work on T cells. So because of the high mutability of the virus, there has long been interest in focusing a vaccine-induced T cell response to target peptides that contain residues that appear relatively conserved when you look at sequences of the same HIV protein derived from virus samples extracted from diverse patients. The idea, of course, is that if a T cell attacks such a residue, then HIV would have to evolve a mutation there, but that should roughly make the virus relatively unfit, because otherwise, why do you see it relatively conserved at that residue? But because of the high mutability and replication rate of HIV, this strategy can be blunted because HIV evolves other mutations, so-called compensatory mutations, that can partially restore the fitness cost incurred by making the primary immune-evading mutation. If you want to describe the mutational vulnerabilities of HIV or other viruses like this, you have to define these collective compensatory pathways that the virus uses to evade human immunity in order to not target them with a vaccine-induced T-cell response. And you also want to define those combinations of mutations that the virus cannot make and still remain viable in order to target them with a vaccine-induced immune response. Or in short, you want to develop the fitness landscape of the virus. That is, knowledge of the ability of the virus to replicate and propagate infection in vivo as a function of its sequence with explicit account of the effect of couplings between mutations. Now, knowledge of this fitness landscape can be very useful for vaccine design because with a vaccine-induced immune response, you want to push the virus off of one of these fitness hills and force it to make combinations of immune-evading mutations that correspond to one of these fitness valleys. And you also want to block these mountain passes that the virus traverses by mutation to go from one hill to another when this one is under immune attack. Now, armed with this fitness landscape, you can design the immunogen, or the active component of the vaccine, to be not made up of whole proteins, as is usual, but made up of only parts of the viral proteome. In particular, you want to minimize including regions which are rife with such compensatory mutations, and you want to maximize regions where multiple mutations are especially deleterious so that the vaccine-induced immune response targets them. And you also want to maximize regions that can be presented by people with diverse MHC genes so that you get broad coverage of a population. It is this very practical goal that led us to try and translate the data on thousands of sequences of viral proteins derived from diverse patients into knowledge of these fitness landscapes. So this work has gone on for a while, and let me just say that the people in the top row were former students and postdocs who have gone on to their own independent careers, and I shall mention these people in context. So the way we do this is the following. Any given sequence of a viral protein can be represented as a vector, Z, because it is a list of amino acids at the various residues of this protein. And we begin with a more modest goal of trying to infer what I call the prevalence landscape of the virus. That is the probability Z, the P of Z, of observing a particular sequence in circulation. Of course, the sequence data contains this information. The sequence data also contains information on the probability of seeing single mutations at every residue, double mutations at every pair of residues, triple mutations at every triplet of residues, and so on. Any mathematical model for P of Z that can recapitulate these mutational correlations is legitimate. The adapting and extending approaches that were developed by other physicists in different contexts, we asked, what is the least biased model for P of C that by construction fits the measured 1 and 2 site mutational probabilities? Then, by exploiting the connection between information theory and statistical mechanics, least bias is interpreted as the maximum entropy model. So let ZK be the kth sequence of a particular viral protein, and the P of ZK is the probability distribution that we seek, and its entropy is given thusly, which we wish to maximize, subject to the constraints that the probability distribution is normalized. The 1 point predicted mutational probability at every site is that which is observed, and similarly for the 2 site mutational probabilities. These constraints are imposed by Lagrange multipliers, and 2 lines of algebra will show you that the formula for P of Z has this very familiar form from Boltzmann's statistical mechanics, where the Hamiltonian is given like so, where the fields and couplings are inferred by fitting the 1 and 2 point mutational probabilities. So large negative values of HI mean that that site is not very mutable without incurring a large fitness cost. Positive values of J imply that mutations at that pair of residues is compensatory, and negative Jij implies that the 2 mutations together are even worse than by themselves. But at the end of the day, what we have here is a prevalence landscape. Now you can argue that the more prevalent strains should also be more fit, but this argument can be complicated because the virus samples that we use to infer P of Z were not viruses growing in Petri dishes, but rather they were viruses that developed in individuals in whom there was a battle between the immune response and the virus. So the virus could have been forced to make a mutation to evade the immune response in this particular person, which actually led to the virus becoming intrinsically less fit. But in this person, it was the most fit strain and therefore most prevalent. Now we seek the intrinsic fitness landscape because we then want to push and pull on it with a vaccine-induced T cell response to corner the virus between getting killed by the T cell pressure or evolving mutations that cripple it. So with these 2 gentlemen, we explored the connection between prevalence and fitness in a number of different ways. And the conclusion that we reached was that for HIV, while the prevalence and fitness are not identical, the rank order of fitness and prevalence is statistically the same. And that's all we care about. We need to know which one is fitter than the other, and so on and so on. Let me show you just one way in which we explored this connection between prevalence and fitness. So this is Eigen's famous equation for evolution of viral swarms. X alpha here is the frequency of the alpha strain, and its time evolution depends upon the fitness of that strain. But because the different strains compete, the fitness of the strain has to be compared with the average fitness of the prevailing viral swarm. And of course, you have mutations out of this strain into other strains and mutations in from other strains to this strain. Now, many years ago, Lauthauser showed that this non-equilibrium dynamical equation is isomorphic with the equilibrium statistical physics of a 2-dimensional Ising model. In this Ising model, each row is a particular viral strain, and the different arrows indicate the amino acids at different positions. And the row next after that is the progeny of this strain, and so on and so on. So any given configuration of this 2-dimensional Ising magnet is a particular evolutionary trajectory. So a path integral description, if you like. And the weight of a particular evolutionary trajectory is given in terms of a Hamiltonian, which in our case, the in-row couplings are the intrinsic fitness of the virus, modulated by the effect of the immune pressure, which is represented as an external field that promotes mutations at the residue where it acts. And across rows, there is a nearest neighbor ferromagnetic coupling, which simply illustrates that if since the mutation probability is less than half, if I have an arginine at this residue, my child is likely to also have an arginine at that residue. Now, once you have this sort of a formulation, you can use all the machinery of equilibrium statistical mechanics. And for instance, one thing that you can do is that this is the actual Hamiltonian operating on this evolutionary trajectory. The Hamiltonian that we infer is quadratic in character, so we can use Feynman bounds to estimate these Kij's and AI's in terms of the parameters that describe intrinsic fitness, the effect of immune pressure, and mutational fidelity. So the formulas we get lead us to some insights. I will skip the details of these formulas and how they lead to telling us what are the underlying biological reasons that lead to the viral fitness landscape and the prevalence landscape being statistically similar for the case of HIV. These biological reasons are the following. First of all, as I told you early on, there is a great diversity of MHC genes in the population. So if you go look at the clinical data, you will find that there is no particular region of the proteome that is targeted by a significant fraction of people. The other important thing is that if I force the virus that I'm infected with to make a mutation that incurs a fitness cost to avoid my immune pressure, and then I infect someone else who targets a different region of the virus, then the original fitness incurring mutation, it reverts over time because it's a chronic infection. And another very important reason is that HIV, unlike influenza, has never been subjected at the population level to classes, a few classes of effective natural or vaccine-induced memory immune responses. So the population of HIV has not evolved in narrowly directed ways to evade the effects of past herd memory immunity in the population. And you can actually see this by looking at the phylogenetic trees of influenza, which looks like a flowing river with branches coming off it, and comparing that with HIV, which I showed you is like a dendromer. So human immunity forces the virus to sample sequence space, but at the end of the day, our analysis suggests that what it does, it basically shakes it and does not statistically change the rank order of prevalence and fitness. Now, the inference procedure, I guess I should call it machine learning to be fashionable, that we have used is approximate, obviously. And also, this sort of analysis is approximate for a very complicated problem. So the only way to establish the veracity of these sorts of analysis is to confront predictions from this model against experimental in vitro and clinical tests. And I'm going to show you snapshots of such comparisons, beginning with comparison against in vitro data. So we can predict the fitness of viruses of bearing different mutations. So for example, the fitness of any given mutant strain is measured by a quantity E, which is simply the Hamiltonian corresponding to that particular strain. And the model suggests that the value of E minus that for a reference strain should correlate negatively with the intrinsic fitness of that strain divided by the fitness of the reference strain plotted in these coordinates. Now, our collaborators can make mutant strains that we predict, whose fitness we predict, and then they can do that by site-directed mutagenesis, and then they can measure the ability of that strain to infect human cells in vitro and grow out in comparison to a reference strain. So the first data I'm going to show you is data where we compared predictions for viral strains with mutations in a large polyprotein of HIV called GAG. And these experiments were then done in collaboration with Tumbi Udungu in South Africa and Bruce Walker at Mass General Hospital and the Reagan Institute. And we made predictions for 36 different strains. And here is the data as dots, and that's the line. And only 27 strains are shown here because for 9 strains, we predicted that the fitness should be very, very low. And they did not grow out at all in the experiments. And those are not graphed because of the difficulty of plotting the log of 0. Now you can see that we are not perfect, but with high statistical significance, we capture about 80% of the data. Here's another comparison with 103 data points for another mutations in another large polyprotein of HIV envelope. This one makes the spike of HIV. And again, you can see we are not perfect, but we do reasonably well. Such comparisons with other HIV proteins are also encouraging. Let me not show you that, but rather show you one snapshot of comparisons with clinical data, which was done in collaboration with Andrew McMichael's lab at Oxford University. Now, Andrew had historical blood samples from HIV-infected patients who had never been subjected to drug treatment. And he had temporally ordered data, samples, so he could sequence the viral swarm as a function of time in these individuals and see how that evolved. He also knew where the T-cell response of these people were targeting the virus proteins. In these individuals, the virus always managed to evolve mutations that evaded this T-cell response. So Andrew asked that of the various locations where mutations could have emerged to escape the T-cell response, using your fitness landscape, can you predict where they actually occurred in these patients? And also, can you also predict the relative times at which escape occurred in these patients? So for over 100 years, people have carried out evolutionary dynamics with fitness landscapes. But to our knowledge, they were mostly done with fitness landscapes that were toy models. In order to answer these questions, we carried out very similar dynamics, but now with the fitness landscape of a human pathogen subjected to real human immune pressure. So the input to this calculation is the initial swarm of viruses and where the CTL responses are acting. So how do we do with predicting where the mutations emerged? Well, we get it right about 86% of the time. Rather than showing you this data in detail, I will rather highlight how our results show the importance of compensatory mutations that the virus uses to evade human immunity, which will also highlight the importance of the sequence background in which the same mutation has to emerge. And this is best done by comparing pairs of patients who target precisely the same region of the viral of one of the virus's proteins. So these two patients target precisely the same region, but in this person, the escape mutation evolves 40 times slower. Now the circle represents the rest of the protein in which this peptide is contained, and the marked residues are those where mutations existed in the virus that infected this person. Blue lines indicate that we predict a compensatory interaction between the pre-existing mutation and the ultimate escape mutation. And red lines indicate that there is an antagonistic effect between the pre-existing mutation and the ultimate escape mutation. That is to say that this mutation makes it even more hard for this escape mutation to evolve. The thickness of the lines are the values of the couplings that we predict, and therefore the strength of this interaction. As you can see vividly in this particular case, this patient was infected with a virus that contained many mutations that were strongly negatively coupled with the escape mutation that had to evolve, and therefore this escape mutant strain was more unfit in this case, and therefore took much longer to grow out than in this patient. Well, how do we do with predicting the relative times? We can only predict time in generations of evolution within these calculations. So here is our predicted escape time in evolutionary generations, and this is the escape time measured in Andrew's lab in the real patients. And again, you see that although there's plenty of scatter in the data in the middle here, with high statistical significance, we don't do too badly. And please note that these are not contrived experiments, but rather natural infection in people. Most importantly, we are very good at separating out regions where mutations are escape mutations emerge very quickly. So these are precisely the regions that we should not be targeting with the virus, and also ones where escape is difficult. And these are sorts of regions that we should be targeting with an immune response. So encouraged by these sorts of data and others that I have not shown you, we decided to try and design an active component or the immunogen for a T cell component of a vaccine, which contains only parts of the proteome. But the trouble is that short regions are very, short peptide-like regions are very difficult to deliver in a way into animals and people in a way that will lead to a strong T cell response. One way around this, we have also done many things comparing with other kinds of patient data that I'm going to skip, and simply say that one way around this delivery problem is to use these adenovirus vectors that I told you in the context of COVID-19 vaccines. But they require that the length of the insert into its genome has to be sufficiently long. So Darius Murakowski, a former graduate student, designed a greedy algorithm which aimed to include as many of the good, that is mutationally vulnerable parts of HIV's genome as we could without including too many unnatural junctions. So let me make this point a bit more clear. So you have a bunch of little pieces. You can't just join them all together and make a whole bunch of junctions, because these junctions are unnatural, and targeting them with a vaccine-induced immune response is not going to be helpful because the real virus does not contain those junctions. That is why you have to minimize these unnatural junctions. So at the end of the day, we produced a 551 amino acid immunogen, and Dan Baruch's lab, who then inserted this into the genome of a adenovirus 26 strain, which is the viral vector strain, by the way, that is being used in Johnson & Johnson's COVID-19 vaccine to deliver the spike protein. And indeed, Dan is the creator of that J&J vaccine. His lab then immunized monkeys with this particular Ad26 containing our immunogen. And this is a small study. 4 monkeys got our vaccine construct, and 2 are controls who got whole proteins as the insert. And Dan has hundreds of monkeys that have been thusly immunized in the past. And this is the strength of the T cell response. And you can see that our vaccine is just as immunogenic as those containing whole proteins after the prime, that is the first shot. Now, notice the scale goes up to 700. Now we give the boost. And after the final boost, look, the scale, the responses have gone up a lot. But again, importantly, the strength of the T cell responses in animals who got our construct is just as high as that in the control that got whole constructs. So because this delivery method is approved for human use, and because we have tested our predictions from the fitness landscape against many different situations, the next step would be to contemplate small trials in a therapeutic context, perhaps in humans. But you know, that requires solving or rather confronting and overcoming many challenges that involve both money and politics. So who knows whether that will happen? And perhaps, while I'm hopeful this will not be so, who knows, perhaps this story will then end where it is today, which is from statistical physics to monkeys. So on that note, let me spend the last few minutes to say a little bit about our work on antibodies. We work both on influenza and HIV, but let me continue the theme of HIV and illustrate our work in that context. So this is a schematic depiction of the spike of HIV proteins, HIV virus. It contains some relatively conserved residues, shown here in yellow, that bind to human receptors. But this region is smaller than the footprint of an antibody. And it is surrounded by highly variable regions, shown here in red. And these regions are protected by sugars, shown here in blue. Now, immunologists have been able to isolate antibodies from some patients that, at least in vitro, are able to neutralize diverse viral strains. These so-called broadly neutralizing antibodies, or BNAbs, bind to regions that contain these conserved residues. The trouble is that these BNAbs usually emerge several years after infection and only in some patients, and that too in small numbers. So they're not very helpful in protecting this person. But nonetheless, they provide proof of concept that the human immune system can evolve such broadly neutralizing antibodies, and suggest the tantalizing possibility that properly designed vaccination protocols could induce such broadly neutralizing antibodies efficiently in diverse people. Now, upon natural infection or vaccination, antibodies are produced by a Darwinian evolutionary process called affinity maturation, which occurs in a short time. I told you that when a B cell's receptor can bind sufficiently strongly to the spike proteins of a virus, it gets activated. It can then seed structures in your lymph node called germinal centers. Herein, these B cells start to multiply rapidly, but also mutations are introduced into the antigen binding region of this receptor at a very high rate. These mutated B cells then interact with the virus or the antigen, which is displayed on other cells in this germinal center. And if the receptor on this B cell can bind strongly enough to this virus, it can pull on it and eat it up, and then chop up the proteins of the virus in here, display it as peptides, and display it in complex with MHC proteins. Now, these B cells that are successful at eating antigen, they then compete with each other to bind to another kind of T cell present here in limiting numbers. And a productive interaction leads to a survival signal. Otherwise, the B cell dies. So you can see that B cells that bind more strongly to the virus will stochastically be more likely to internalize more antigen, which will then likely be able to, more likely to display more peptide MHC on its surface than its peers, and so more likely to get this interaction, survival and interaction with the T helper cell. Now, a small fraction of the positively selected B cells are let out as memory B cells and plasma cells, with these guys secreting the receptor insoluble form as antibodies. But the vast majority are recycled for further rounds of mutation and selection. So you see that as time ensues after infection, due to this mutation selection processes, the antibodies that emerge bind more and more strongly to the antigen and thus become more potent. This problem has been studied a lot, both by experiment and competition, continues to be studied, but mostly in the context of immunization, that is vaccination, with single model antigens. Our interest is to see how we can trick this Darwinian evolutionary process to make specialists, generalists, that is broadly neutralizing antibodies, rather than specialists, that is strain-specific antibodies. So you should be able to see that the induction of these broadly neutralizing antibodies will require vaccination with more than one variant antigen, because if you immunize with just one strain, this Darwinian evolutionary process will make a very good strain-specific antibody. So you will require to immunize with many variants which share the conserved residues, but their variable parts are different from each other. So which raises many new questions. Which variant antigens? How many? What should be the mutational distances that separate the variable regions of these different variant antigens from each other? Should I give them all simultaneously as a cocktail of variant antigens, or sequentially, or any permutations thereof? So the answers to these questions are drawn from a very large space of possibilities. And it may be difficult to find the golden nuggets that we seek by randomly sampling the space in experiments with monkeys. Now, a mechanistic understanding of this affinity maturation process would provide guidelines for the choice of answers to these questions. But as I said, until very recently, affinity maturation had largely been studied in the context of single model antigens. So how it occurs when there are multiple variant antigens or selection forces is not well understood. So therefore, we developed some computational models to help provide some insights into this fundamental problem at the intersection of evolutionary biology, immunology, statistical physics, and learning theory. And our goal and hope, of course, is to use those insights to guide immunization strategies. Given the time constraint, I will not describe our results over the many years, which have been obtained by work with many junior scientists who were in our group, who have gone on to their own independent careers, either in academia or more lucrative pursuits. And it has involved collaboration with a number of biologists and also physical scientists, such as the ubiquitous Professor Kardar, whose influence on my work cannot be overestimated. And the way we do this is the following. The B cells get activated. So simulation, the way the computation works is that B cells get activated, they replicate and mutate. There's affinity-dependent selection. A small fraction of the positively selected ones are let out. The others are recycled for many rounds of mutation and selection. The simulation or the calculation comes to an end when all B cells die, that is extinction of the population, or all B cells exceed a threshold, that is a proxy for the fact that they've eaten up all the antigen, or after a maximum time, that is the antigen has decayed. And we interrogate such models using either stochastic agent-based computer simulations, Fokker-Planck equations, or master equations, and their output is the probability of obtaining BNAbs under different immunization conditions. I will just summarize our results and no more in 2 slides. The first result that we have is if you give these variant antigens as a cocktail, then there is extensive B cell death and extinction of the population, so you don't get antibodies in large numbers. The reason for this is illustrated using this cartoon. So here is a depiction of 3 variant antigens that share the red conserved residues, and the different colors indicate that they have different variable parts. And imagine that in one round of mutation and selection, this B cell was positively selected by this antigen. It comes back, it mutates, and then goes for selection again, and interacts with a different antigen as the selection force. In the early stages of affinity maturation, the affinity of the receptor for the antigen is not very high. So the chance that it has developed strong interactions with the conserved residues is low. Therefore, the chance that this will bind strongly to a different antigen is also low, and death is the likely outcome. These variant antigens present together are conflicting selection forces that can lead to extinction. If we vaccinate sequentially with the same variant antigens, then we had predicted that the evolution of BNAbs will be more likely, and that has been tested in animal experiments. What do we mean by sequential immunization? Well, when we give the first variant antigen, memory B cells form by adapting to this selection force. When we give the second variant antigen, the existing memory B cell population is thrown off of steady state or equilibrium and has to adapt again. And an important question is, to what extent should we keep throwing the memory B cell population off of equilibrium or steady state in sequential immunizations to optimize the evolution of broadly neutralizing antibodies? And in recent work with 2 postdocs, Kayla Springer and Raman Ganti, we have obtained the following thoughts. If the distance, mutational distance between the second and first variant antigens is too low, then after the second immunization, you will get a lot of memory B cells surviving because it's not so different from the previous one. And at the same time, though, they will acquire very few mutations. So for the third immunization, when you really have to push it to evolve these broadly neutralizing antibodies, most of these B cells will have dead-end trajectories. If you have too high a mutational distance between the second and first variant antigens, all the B cells will die. And of course, then you will not get BNAbs in subsequent immunizations. So we find that there is an optimal mutational distance between the first and second variant antigens, which leads to the right kind of B cell diversity and enables many paths to success in subsequent immunizations with variant antigens. Now, there is also an optimum for subsequent variant antigens, but these optima drive the existing B cell population even further from equilibrium than that in the previous immunization, so that together you can learn how to evolve these broadly neutralizing antibodies. So let me just end by saying the following. The affinity maturation is a non-equilibrium dynamical process with two very strong attractors, the value of depth and strain-specific antibodies. And there is a narrow ridge line that connects you to this promised land of broadly neutralizing antibodies. And we are finding that there may be optimal sequential immunization strategies that can convert this narrow ridgeline to a broad stairway to heaven. So with that, let me stop and say only this, that we have also been doing quite a bit of work on T cells in COVID-19, but I shall not describe this here today and stop here, and I'm happy to answer any questions.
I guess let us thank Arup.
Thank you.
Should I lead the questions, Robert?
No, go. Yeah, you should lead the questions. Absolutely. Yeah, and then we'll chip in. And I think Arup, maybe you want to stop sharing the screen so at least we go. Except if you have certain things else to show, but definitely. Thank you so much. It's been wonderful. So, Mehran?
So thank you, Arup. I guess there is a lot of interest in applying the methodologies that you described to the SARS-CoV. And I was wondering whether there is enough sequence data to construct a fitness landscape, or whether it's also possible to use in vitro experiments to artificially deduce a landscape.
Yes, that is true. But first, let me say that why would you be interested in developing landscape? It would be under two circumstances. And one is that, because at the moment, SARS-CoV-2 is not evolving many mutations. But in any case, one way might be that if we do get these successful vaccines, then much like influenza, we may get strong selection forces that direct the evolution of SARS-CoV-2 to evade those viral vaccine-induced responses. And there are some papers, there is one paper in the literature right now that seems to suggest that may be true. So that's one way. The other, which actually I am more interested in, is that can we develop a fitness landscape for coronaviruses, and rather than SARS-CoV-2 alone. This is a pie in the sky idea at this time, but perhaps then we can learn, are there regions of the fitness landscape that suggest that would be difficult even during zoonotic shifts from one animal, the viruses that grow in one animal to another, even during those zoonotic shifts might be preserved. One suggestion in a paper, again, I think it's only on bioRxiv, is that children are targeting with their antibodies a region near the base of the spike protein of SARS-CoV-2, which is common with other coronaviruses that cause less pathogenic diseases like common colds and things of that sort. Can we combine analysis of sequences and structure, such as people have done with machine learning, to try and learn where those swaps would be, and then use all the approaches that I described here to try and design immunogens that could target them, either the antibodies or T cells. In some of our T cell work, we have also seen with our fitness landscapes that, and others have seen this too, that there are regions that are immunodominant in humans with particular genomes that are common between the low pathogenicity coronaviruses and this. So yeah, it's exciting to think about these questions. Your question as to whether there are enough sequences is something that we are trying to evaluate right now, and we'll see what happens, whether this is worth doing, and if it's worth doing, how to do it.
Yes, there is a related question which says, is it possible to include the structural information apart from the sequence information in building the statistical model to improve predictions?
Yes, so there are two threads of thought here that I need to combine. One of them is that, by the way, we have in our past work seen that some regions are very difficult to mutate in all sequence backgrounds based on our fitness landscape. And we have seen in the past that they do arise because of structural constraints in multi-protein assemblies or within the same protein. That is one thing. The second is that Bruce Walker, along with his postdoc, has tried to do network analysis, has done network analysis of HIV protein structures, and has also come up with some mutationally vulnerable regions based on that. And that is what I was saying in answer to your previous question, that combining such structural analysis with this sequence analysis together might lead us to better ways of thinking about these fitness landscapes. And certainly something that I would want to do in the context of SARS-CoV-2, for example, not just that.
We have a question from Sharon Glotzer regarding mutations and SARS-CoV-2. What do we know about the mutation rate of this coronavirus? And do we need only worry about mutations in the spike protein, since the business, which is the business end of the virus, are the new vaccines likely to fare against future mutations in the spike protein?
Yes, so there are, hello Sharon, good to hear from you, at least by proxy. So, so there are many questions here. First of all, what is the mutation rate of SARS-CoV-2? So RNA viruses mutate a lot, but SARS-CoV-2 is not mutating much. Why is that? That's because coronaviruses have among the longest RNA genomes of RNA viruses. If it really had the normal mutation rate, they would go past the error threshold very, very quickly and die. So coronaviruses have evolved another protein called NSP14 that it brings with it, which acts like a proofreader of sorts and prevents too many mutations from occurring. But now the question is that if we put selection force through very effective vaccines deployed worldwide, will it be still able to evolve new mutations that still maintain replicative fitness? In that case, it would become like flu. And again, as I said, there is one paper that suggests that this is possible. What will happen to the old vaccines? Well, that will depend upon how well SARS-CoV-2 can evade this. We don't have information on this, but this is something that in vitro studies are beginning to explore. The other question you had was only the spike protein. Well, this is not, the immune system did not evolve T cells for fun. So there are two aspects to this, that, the antibodies can prevent infection and give you sterilizing immunity, which happens very rarely. But the T cells clean up any infections that may happen. So that's the thing that as more data comes in will become clear, that whether adding T cells to this vaccine component in the future might make it even better, because it will also clean up any messes that happen. But we'll see as data evolves.
Next question from Alan Perelson. In the experiments done by Dan Baruch, were the monkeys challenged with a diverse viral quasi-species of SIV that might reflect the person needs to be protected against?
Alan, no, I'm sorry if I misled you. The studies so far that I showed and unpublished with the monkeys were just an immunogenicity study we have not yet done the challenge study. What we do know is that we are mounting strong enough T-cell responses as whole protein antigens, and that they are targeting the mutationally vulnerable regions. The challenge study has not been done yet.
This is an interesting question. Since the SARS-CoV-2 spike is so much more conserved, why is this strategy of 2 vaccinations that is being proposed for COVID-19? Do you know why that is?
Yes, so typically when you vaccinate, just as I was trying to describe in the second part of my talk, perhaps too quickly, is that what the prime, the first shot does, is it gets some memory B cells activated and mutated to get better for the virus. And then you give a second shot, which is the booster shot. And what that does, it takes the existing memory B cell population, and affinity maturation can occur again, leading to more evolution to make it even fitter. And at the same time, also, the previous memory B cells can also expand outside of the affinity maturation, which again expands the higher affinity ones. So that is the purpose of the second shot. But I should say that the J&J vaccine with the adenovirus carrier vector, that is being tested as a one-shot vaccine, because the monkey studies that corresponded to this suggest that the antibodies developed were sufficiently potent and large in numbers with one shot. And they got similar sort of results from phase 1, phase 2. So that is a one-shot vaccine, which would make the logistics much more simpler than a 2-shot vaccine. So anyway, the question, why we boost? We boost in order to get another evolution and expansion of the higher affinity cells.
So can I maybe just add a follow-up question to this? If you compare the 2 classes of vaccine, adenovirus-based vaccine and RNA-based vaccine, what do you think is the long-term better robustness of the vaccine, just because in terms of evolutionary pressure?
We don't know, because one thing that people forget, I don't know if people forget, one thing I wanted to say is that RNA vaccines and adenovirus-based vaccines with a genetic insert are both genetic vaccines, okay? And no vaccine in the past using either RNA or adenovirus has ever been deployed in large scale. An RNA virus has never been even licensed. And the Ebola virus using Dan's adenovirus vector was recently approved for Ebola in Europe, and that's it. So we don't have experience about durability of vaccine responses with either approach, or for that matter, any approach at this time. So,
can I ask a question? So I have a question about your last part of the talk, where you talked about using different viral antigens to target the disease to improve the immune response. And so would this be useful in flu vaccines? Annual flu vaccines are given where they take a bunch of strains, but I'm not sure who chooses the strains, how they're chosen, but could you optimize this in some way that would make it more efficacious? Or
well, I think a different way to think, a slight variant of your question is what I can answer, which is that one can also think that, I didn't talk about this work, but the hemagglutinin, the flu spike, also contains some relatively conserved residues, which are preserved across strains. And so there has long been efforts directed toward trying to see if by immunization strategies, you can shift the immunodominance to target those residues upon vaccination, then you have a universal flu shot. Yeah. And so recently with Dan Dingwood's lab, we published a paper in Cell Systems, which takes a very tiny step toward that, contributes to that goal. The trouble is that with flu, the spikes are placed very, very closely. And one of the very good conserved residues is in the stalk of the spike. And so accessing that is difficult. So how do you change that immunodominance hierarchy? We have worked with Daniel, a young scientist, professor at Harvard Medical School, who did the mouse experiments and has the antigens which display these on nanoparticles. And so you can play with the the sizes and the spacings. And also in influenza, you have to worry about the fact of what have I been exposed to in the past. And so you can make these next antigen that you give on these nanoparticles to be different from past exposures so that the ones that target these get amplified. Yeah, so we have paper came out about a month ago, I think, on this.
And I have one more question, which is that in your first talk, did you take into account different MHC types in?
Yes, we did. Very much so. But the monkey study, we also had to account for the different outbred MHC types of monkeys. But as we now move this forward, we are going to think about those issues more even.
Do the different strategies depend on MHC types?
Yes, because you have to find no, that you cannot do for a vaccine, because then you don't want to make personalized vaccines for something like HIV. So what you want to do is have regions in your vaccines that have immunodominant peptides that can be presented by a broad coverage of people, and that's what we are doing. And that is why we have another effort, which really led to our work in COVID-19, to try and predict immunodominance given a genotype of a person.
Mm-hmm. Okay. Thank you. Yeah.
So maybe while Mehran is looking for some others, I think there is another interesting question and it's more generic. Does intrinsic landscape, fitness landscape even exist? Wouldn't that depend on each person and their immune system in some way? So, in some way is how this population-based vaccination obviously has to do versus something that's more personally tailored. Is there any way to go into more personalized vaccination any time?
Well, for therapeutic vaccination, which is one thing we are exploring with some of the T-cell approaches I described, in advanced nations like ours, we can type the MHC, which is called HLA typing of the person, and then try to see whether your vaccine construct that you have contains things that are immunodominantly presented by person with that HLA. If your vaccine construct does not, or this person's virus has already mutated that region, then that person is not a candidate for this vaccine. And with Boris Job's lab, we are trying to explore that particular route. So that what we're trying to do is get the immunodominance patterns.
Yep. Thanks.
So I have a question regarding immune system response by T cells or B cells. How do you sort of decide by vaccination which part of the immune system is best suited for targeting the pathogen?
So you have, for a prophylactic vaccine, you have to have an antibody component so that you can plug the virus before it infects cells. And the T cell component can attack all the infected cells. So without an antibody response, to have a prophylactic vaccine would, I think, be very challenging. The vaccine so most vaccines employ an antibody response. The vaccine, a well-known vaccine and very effective vaccine that employs a very strong T cell response to go along with it is all vaccines have some T cell response, or many of them do, but the yellow fever vaccine, which is very good, is very much dependent on a T cell response as well. So you cannot, but in a therapeutic context, the one I was describing, you have a person who is infected with HIV and you take them off of drugs, give them a vaccine, then take them off drugs if they can control the virus. Their T cells are the key component, along with potentially. People are also exploring giving passive, broadly neutralizing antibodies into people and seeing if you can keep the virus under control instead of the drugs. So anyway, for prophylactic vaccine, I think you have to have an antibody component, and T cell component will help. But for therapeutic vaccine, you have to have a T cell component, I think, as well as an antibody component. The T cell component being very important there.
And what determines the length of time over which immunity remains in the body?
And nobody knows. So in some virals, and so this is, but by the way, this business, I mean, I saw today, finally, the New York Times had an article, this business about the antibody levels are declining. You know, the immune system works in the way that they have memory B cells that can rapidly ramp up production of antibodies. So just looking at antibody levels is not the question, but it is this is not known. For some infections, the immunity lasts for the lifetime of the person. For others, such as common coronaviruses, it obviously does not last very long. And for SARS-CoV, where long-term data is available, the antibody responses decline quickly, but the T-cell responses stay on for a very long period of time.
Would it not depend on severity of disease?
For sure.
Yeah,
That is for sure. But on the other hand, people with similar severity of disease in two different types of infection lead to different lengths of immune memory. So
because of the way the immune system responds to different kinds of
right, and I think that one of the challenges that I think physical scientists along with biologists can help explain in the future is to understand innate immunity. Our understanding of adaptive immunity has gone faster than our understanding of innate immunity, and that plays a very important role in vaccine responses and other responses, but also causes trouble.
So maybe just and maybe we should slowly wrap up. But like one, I think clearly you mentioned, and I think this COVID-19, I think developments will definitely, for sure trigger a lot of search for general coronavirus vaccine, as you mentioned. That would be the ultimate goal. So how far do you think we are? Do you think that this is like providing a lot of new information, or is just not the landscape of possible coronaviruses is not broad enough to really sample, the landscape sufficiently? Do you think, what's your thoughts? Are we any closer with this, large amount of data that's being generated, or still just at the beginning to embark on that?
I think we're in the beginning, because we also, I think if you want to make a pan-coronavirus vaccine, we also need to collect a lot of data from animals reservoir. So that, I think, will be a vital element as well, not just human coronaviruses. So I'm sure that some of that will start to happen now. And, similar things need to be done for influenza, because we shouldn't focus just on coronavirus. I mean, there have been more pandemics because of influenza. And there too, there is an animal reservoir. And the animals there, we interact with as human population a lot, birds and pigs and so forth. So I think we should also focus also on influenza.
So I guess like physicists should drive what data should be taken, so can help, like I think as you really showed, to drive the next generation of developments.
Yeah, and a lot of physicists are working on very important topics, not just on that, but I mean, if you look at people who have gotten really into the epidemiological models and so forth from physics, it's been very impressive. Okay.
I think Arup would just like to thank you so much again. It's been really wonderful journey that you showed us, and I think all the parallels with vaccine. I think everybody learned much more about where we are and what to expect. So thank you so much. Thank you, Mehran, again also for moderating and helping, and Jan obviously to help running this webinar. So with that, I would like to close and then invite everybody in 2 weeks when we're going back to aerosols. So another obviously very important topic in this times. Thanks again, everybody.
Thank you.
Bye.
Thank you.
Bye-bye.